This morning, we would like to invite everyone to remember, most of us, probably all of us, are, as Adventist Christians, are aware to one degree or another the basic principles that were given to us over 100 years ago by Ellen White regarding health. It's only been really in the past, especially 20 years, but even before that, when I was in medical school in the late 1970s and early 1980s, a lot of the material that we had been told hadn't really been validated scientifically, but now most of it has, and we're privileged today to have a speaker who's done a big part in bringing a lot of the scientific evidence into the fore. He, Dr. John Kelly, got his undergraduate degree in biochemistry and molecular biology at Shepherdstown, West Virginia. He graduated summa cum laude as a McMurrin scholar. Got his medical degree, MD degree at Loma Linda University in 2000, and it also got an MPH degree in 2002 with honors. He finished his preventive medicine residency at Loma Linda in 2003, and became board certified in preventive medicine in 2004. He received the AMA's Excellence in Medicine Award in 2004 for leadership and involvement in founding, being one of the founding fathers of the American College of Lifestyle Medicine, and was the first president of that organization, which has grown enormously. After training at Loma Linda, he accepted appointment at Loma Linda as an assistant research professor in nutrition in the nutrition department at Loma Linda University with the Lifestyle Research Initiative. He's been involved in lifestyle research over the years. He's been medical directors of several lifestyle centers in Maryland, Virginia, South Dakota, Michigan, and now serves as a president and director for Oak Haven Health and Country Life Natural Food Center, where he conducts clinical training in intensive lifestyle medicine. He co-authored the Lifestyle Medicine Board Review for the American Board of Lifestyle Medicine, which gave its first board exam in 2017, and actually has been involved in teaching both patients and professionals, healthcare practitioners, both physician and non-physician, and is truly a lifestyle medicine expert. He enjoys family life, Christian ministry, agriculture, hiking, camping, among other things. And today we're very privileged to have Dr. Kelly with us here today. Dr. Kelly?
Thank you so much for that kind introduction, and hopefully we can see how genetics is really providing a scientific basis that I believe is going to and is lifting lifestyle change, or what Adventists would have called 100 years ago the health message, into prominence in medicine. So thank you again. Let's just begin our little journey here. So I have no conflicts of interest that I'm aware of or disclosures to make for this presentation. I want you to imagine with me for a moment a diet that could induce beta cell neogenesis, a way of eating that could restore beta cell function in pancreas without using stem cells and the risk that has posed. You may be aware that stem cells sometimes seem to have a mind of their own and don't always do just what we would like them to do when they differentiate. Well, you don't have to imagine, because this has been done in a mouse model of diabetes and some parallel work done with in vitro human pancreatic cells. Actually, this was published almost 3 years ago now, I guess, in Cell, the leading journal for this kind of thing. It's called the Fasting Mimicking Diet. The title was Fasting Mimicking Diet Promotes NG3-Driven Beta Cell Regeneration to Reverse Diabetes. The NG3 and some of these other ones, I won't go into details, but these are just epigenetic factors. These are molecules, most of them proteins that are in our cells that control gene expression. So what they found in mice was that a 4-day fasting mimicking diet induces a stepwise expression of epigenetic factors. The names there are printed, but they're not relevant to our main point. And so these epigenetic switches or factors cause the generation of insulin-producing beta cells, and they remarked in the research that it resembled the process that we observed during pancreatic embryonic development. These cycles restored insulin secretion and glucohomeostasis, both in type 2 and type 1 diabetes mouse models. The type 2 is wonderful, but the type 1 is phenomenal, I would say. And really, the key point here is, of course, is somewhat as one of my friendly critics has said, John, we are not mice. That is true. But the fact that this can be done in mice is extremely promising about the potential for what might happen in humans in the future. So in human type 1 pancreatic islet cells, fasting conditions produce similar activity. Those acronyms there are epigenetic factors that induce the expression of these same epigenetic factors that we saw in the mice and insulin production. So basically what this slide is saying is the researchers were showing the rationale for why we would expect similar things may happen in human pancreas. So here's their quote from the paper. These results indicate that a fasting mimicking diet promotes the reprogramming of pancreatic cells to restore insulin generation in islets from type 1 diabetes patients and reverse both type 1 and type 2 phenotypes in mouse models. So it's very promising. Our learning objectives are to discuss the function and operation of the gene expression. This is basically the epigenome. Then to discuss the epigenetic link between lifestyle and gene expression, and then discuss key studies of plant diet and disease reversal mediated by epigenetics. If I might just say in plain English what the main point is of this presentation, and I give it frequently to my patients in lifestyle medicine treatment programs, because what it does, what we're going to review here is the science as physicians and pastors, whoever, I don't know who all we have with us today, but I geared this for CME. So we're showing the scientific evidence that lifestyle changes, diet being one of the prominent ones, can literally change the gene expression in human beings in such a way to produce or remove chronic disease. Well, it turns out that epigenetics really has changed almost everything. If you are, any of you who may be students of this, deep students will realize that the research that was being done primarily in cancer research is where we discovered the gene switches, the things controlling gene expression. All this stuff that used to be called junk DNA. In fact, it was called junk DNA when I took my training in biochemistry in the 90s, is not junk at all. It is very active in controlling the expression of the genes in the other 2% of the DNA. Our DNA is no longer our destiny. Lifestyle choices, particularly diet, as we'll show you this, greatly affect gene expression. It turns out that epigenetics controls almost everything. In fact, it may be that it does control everything. Even the way medications or surgeries, even psychiatric treatment, it's astounding where almost nothing seems to happen in the human body that does not affect gene expression. And as I mentioned here, you'll find this, I think, quite remarkable. I know I did, that epigenetics can control the cell type and the age of cells. We'll look at that briefly. So lifestyle change is necessary and may be sufficient to optimize our epigenetics. So therefore, lifestyle medicine is destined to become a cornerstone of medical treatment. You may be aware that now the NIH does not do a study involving the DNA without looking also at the epigenome, because the gene switches are just as important as what the genes are. And so evidence-based treatments of the future are going to be these things that we're all quite familiar with as Seventh-day Adventists. Basically, the New Start principles are here. So let's just take a look at a couple of amazing things. Here, published in Cell over 10 years ago, over a decade ago, they knew that a combination of 3 transcription factors were capable of reprogramming dermal fibroblasts, okay, skin cells, directly into cardiomyocyte-like cells. And the cardiomyocytes that were produced from the skin cells, so they were taking fibroblasts and activating these 3 transcription factors in such a way that they turned the cell from a fibroblast into a cardiomyocyte, expressing cardiac specific markers, a global gene expression profile that was similar to cardiomyocytes, and they were contracting spontaneously. In other words, these were cardiomyocytes. So here's a picture of mouse embryo fibroblasts, which then were transformed directly into cardiomyocyte-like cells. They are, of course, very cautious in the way so we call it cardiomyocyte-like. But if we were to see these in a pathology slide, they would be classified as cardiomyocytes. They transplanted those transformed fibroblasts into mouse hearts after this transduction process, and they differentiated. This shows that you can take differentiated somatic cells and with epigenetic changes, turn them into a different type of cell. Again, what is the point? The point here is the power of epigenetics. It's hard for us to grasp this, the amazing ability of cells to transform from one type into another simply by changing gene expression. Another little insight here. This was published in 2013 by Andrew Feinberg, one of the leading experts from Johns Hopkins on epigenetics. And what they've discovered is that aging, cell aging, is actually imposed upon cells. This is not a time process. It's not based on chronology. It's actually based on epigenetic switch changes. So the typical phenotype of age or phenotypic aging is not monolithic. The phenotype changes of aging are produced by epigenetic programming which overexpresses some DNA, some genes, and underexpresses other. And the important point is that this is programming that can be changed. And they have done this in the laboratory. So here we just see an article from Bioinformatics of 2009 showing here genes on the left that are overexpressed as part of the cell aging process. And I want to emphasize we're talking cell aging here, not body aging. There's some differences. And then on the right-hand side is the underexpressed, and they've been able to they are discovering the epigenetic factors that can reverse these effects. And that's how, in essence, that's what you do when you turn a differentiated cell into a stem cell back to its pluripotent state. That's what they're doing. They're changing gene expression to make the cell revert back to an earlier phase in its existence. Here's another little illustration, again, of the power of epigenetics. That's what we're working on right now, is understanding and the ability and the power and the extent of which epigenetics controls things. So what we have here is some course, I acknowledge I'm a bit of a biochemistry nerd. I love it. And this is some elegant research that was done on yeast. And yeast, it turns out, of course, does not have lungs. It doesn't have a circulatory system. It's a single cell organism, but it has to have ox-redox cycles. In other words, it has to have cycles where it burns oxygen to produce energy and where it exchanges carbon dioxide for oxygen. And that's called the ox-redox cycle. It turns out that that cycle that occurs in yeast, which we would think of as a sort of a breathing cycle or metabolic cycle, is driven by epigenetic changes. There are switches in the yeast cell that are turning on genes and turning off genes in a cyclic manner, as demonstrated here. And without going into the details, what these charts show and the things that they've done is they've proven with chromatography, with messenger RNA analyses, that the gene switches are indeed driving this ox-redox cycle in yeast. So I want to, this is a medical, more or less, presentation. I want to start with a couple of case histories, if I might. So as was mentioned, I worked in the Lifestyle Research Initiative, and the main thing that probably of significance I did during that time was we did a $2 million research project in the Marshall Islands, where they have a absolute pandemic of type 2 diabetes. About 40% of people over 18 years of age have type 2 diabetes in the Marshall Islands. It's just destroying their health system, because 80% of the admissions to the hospital are avoidable complications of type 2 diabetes. So we did some research. Some of you may recognize that gentleman there. That's Wes Youngberg. He was in Guam at the time, and he helped us out. Another person you might recognize is here, Brenda Davis, in the compounding pharmacy, I call it, the kitchen. And then at the bottom, we have the exercise room. That was not for, that is not what we taught them to do in the Marshall Islands. We did not teach them to exercise on machines. We used the machines to evaluate their fitness. We taught them to do things they could do without anything complicated, and that was primarily walking and swimming, since they have a lot of water there. This was the results of the first intervention group. We had, I believe, 16 people in that cohort, and there was a 10-day intensive intervention. So we were feeding them all 3 meals each day, except on weekends. They went home. And you can see the regression line there is basic linear regression. And what it shows is that during those 10 days, there was a 74 milligram per deciliter drop in fasting blood sugar. I've shown some of these in slides like this in some conferences where there are experts, diabetologists, and what did we do? They want to know if we actually used insulin pump, or how did we get that kind of a drop? The fact of the matter was, the 2 people in this group that were on insulin, we had to stop because we were using something more powerful than an insulin pump. We were using diet and lifestyle, simple changes. At the time, we did not realize that what we were doing was changing gene expression. This was done in the 2006-07 age, and that was when epigenetics was just barely starting to become, common knowledge. Another case history. This is from an outpatient lifestyle center I ran after I left Loma Linda in 2006. For about 5 years, I ran a outpatient lifestyle specialty clinic, and using the CHIP program. There's a little here, a little TIFF showing a simulated echocardiogram, because that's what is going to be involved in this story about this patient. So Charles, yes, a relative, and I have his permission, a brother, here to share this information. He was retiring from his very good job with very good insurance coverage, and he thought to himself, I think I ought to get a complete thorough checkup before I retire. and go on Medicare. And so he had a very thorough internist. I'm impressed with what she discovered. She discovered that there was an unusual signal on the EKG at the top end of his treadmill test. And he was quite fit, actually. I think, if I'm not mistaken, he was up around 11 or 12 METs before he stopped. But she saw this change in the EKG, sent him to a cardiologist, who Dr. Spady. And Dr. Spady did an evaluation with a stress echo. And as he says here, it suggested inferior ischemia. And so the plan that he was recommending was to have an angiography with intervention if indicated. And of course, any lay persons, that means a stent, was what he was suggesting. We will look for the blockage and put a stent if we find one. Well, Charles was a reader, and he did some reading and decided that he wasn't sure he wanted to have a stent placed. And so he had a brother that was into lifestyle medicine. He calls me up and says, I want to do lifestyle. I don't want to have this stent. And I said, Charlie, there's 9 of us kids. You know, if you die and the rest of the family thinks that I killed you by not letting you go get your stent, I'm going to be running for the rest of my life. I says, I'm only going to go along with this if you're going to make some serious changes. And I know my brother, at the time, he was not a Christian. He had not lived as a Christian. And frankly, he was like I used to be before I was converted. He was pretty hedonistic. If it felt good, he did it. So he says to me, he says, oh, I'm serious. He says, I'll become a vegetarian, like that was a life sentence or some sort. I says, well, I know, but beer and pretzels are vegetarian, so I'm not sure that's going to fix the problem. Well, anyway, we had our serious discussion, and I really made it hard. And he said, no, I want to do this. I've got to do this. So he came to our lifestyle center, actually stayed with us in our guest room, went through the program, and went and got another stress echo. It was almost 2 years later. And this was Dr. Austin, a physician that I knew from the area in Virginia here. And so what was the plan? This echo was considered negative for ischemia. And he said the patient's done well with lifestyle. The plan, he'd continue with diet and exercise and aspirin, baby aspirin. So that was quite a remarkable, in fact, talking with Dr. Austin later, he said to me, I've never seen this. To be honest with you, I wonder if that first echo was an artifact. Well, of course, Dr. Spady wasn't very favorable to that idea, that his study was an artifact. So this was, but I share this because this is one of the most prominent, clear cases I have of proof of reversal of cardiac ischemia from lifestyle changes. And if you want, it's interesting, take a look at this. This is from New England Journal of Medicine, 2009. And what this was a study actually looking at, well, look at the title down at the bottom, endoscopic versus open vein graft harvesting in coronary artery bypass surgery. So basically the physicians had noticed there seemed to be a difference in the blockages in the restenosis of a bypass graft, depending on whether they harvested the vein with open excision or laparoscopic. And so sure enough, when this study was published, you can see that the failure rate, this is a 12 to 18 month graft failure rate, was about 38%, or it was 38% for the grafts that were, the saphenous vein was, open excision, versus 47% for the ones where they had harvested laparoscopically with a combined rate of 43%. So 43% of the grafts failed within 12 to 18 months. Now, folks say, might ask, why is a lifestyle medicine physician even looking at articles about, failure rate of grafts? Well, the reason is because we could make this go to zero practically if we would do lifestyle change along with the bypass. So I want to compare that, for example, that study to this one here that you've seen, if you've seen any of Caldwell-Esselstyn's stuff. This is his classic angiogram that they did of the doctor who worked with him that couldn't take statins, but he had blockages. So the left-hand side was the LAD before he had this 32-month plant diet intervention program, and on the right-hand side is afterwards. And while these images are not perfectly clear, there's clear enough that we can see major changes in the blockages, stenosis in that left anterior descending artery. But you know, every time that I share these stories, these case histories and the things, I always have some people in the audience who say, yes, but you know, John, I've just got bad genes. I mean, it's in the family. It's both sides of my parents and the grandparents and my brothers, everybody. But you know, there's something that we need to keep in mind. We don't just inherit DNA from our parents. We inherit cookbooks. And in fact, there's many a lady who has, after getting married, has had to learn to cook a few things that her husband's mom or grandma used to fix. So anyway, it's interesting. If you haven't seen this, this was in the January 2010 issue of Time magazine. And why your DNA isn't your destiny. And the subline was, the new science of epigenetics reveals how the choices we make can change our genes and those of our kids too, which is actually seems unbelievable. So stick with me here. How can my lifestyle affect the genes of my kids? So a little history, actually, because what I'm going to tell you is most of it already. So let's do a little review. Here's Watson and Crick. They're receiving the Nobel Prize for their discovery or evidence that proved that deoxyribonucleic acid is the molecule of inheritance, the molecule that contained the information, the genes. Now, just a little thing while we got that graphic there on the screen, it almost looks like, to me, it looks, DNA looks a little bit like the double helix, like a twisted ladder. If you can imagine that you had a ladder with rungs on it and you sort of twist it. But if you look more closely, or if you know, actually the rungs are not attached. There's these base pairs is what each one of the rungs on this twisted ladder, that's called a base pair. And they don't actually have a covalent bond. They are simply held together by hydrophobic forces. And that's why, of course, they can be separated when you transcribe a gene. So that's going to be important in the next slide, because when they did the Human Genome Project and we sequenced the human genome, we found 3 billion of those base pairs. Let me just go back. 3 billion of those rungs on the ladder, if you will, in the human genome. There was about 25,000 or so individual genes and millions of variations in the base pairs. And of course, some of those cause a disease, but most don't. And most people are aware of these variations mainly because we use them for this DNA testing that's can tell one person from another with greater accuracy even than fingerprints. In fact, many people have either been convicted or taken off of death row because of these variations in the DNA and the DNA testing. There's, of course, Dr. Francis Collins, who led that project and is now the director of the National Institutes of Health. So here's an obvious, the most common example, I would say, of where a difference, a variation is harmful. This is sickle cell disease, and a single base pair substitution causes the amino acid valine to be in the hemoglobin instead of the glutamic acid. And this causes the cell on the left there to sickle when the pH is gets low, and that cell cannot circulate through the blood vessels. It will not fold and pass through the tiny vessels, and it causes ischemia, and it can cause death, as you know. So Another little review of things that we already know, really, from our biology, and that talking here about some mammals. Where do we come from? So here's a picture of a hand holding three little creatures, almost looks like some kind of chinky pen or something. But this is actually, the question here is, where did these come from, these little hedgehogs? So we know biologically that an ovum from the mother was joined by a sperm from the biological father. Each one of those cells were actually only had half of the chromosome. And so they had to come together. And in the case of humans, then they have 23 pairs, right? And that's called a zygote. That zygote then starts to do what it does best, which is divide, and makes two cells. And each of those become two cells and so forth. And 9 months later, if everything goes well, praise God, we're born with trillions of cells. And they all came from that first cell. So the question I have for you is, if every cell has a copy of the same DNA, how does it produce such different tissues? Some of it makes an ear, some of it makes a toe or brain cell, all coming from the same exact DNA. Well, as Dr. Whitelaw says, we don't inherit DNA. What we inherit are chromosomes, and chromosomes are only half DNA. The other half is made up of protein molecules, and these proteins carry the epigenetic marks, switches, you might call it, information that drives and controls gene expression. So stop a moment and think. So then when I was born, when you were born, of course, I had the 50% of the chromosome that were the genes from mom and dad. But the other 50% of the chromosome that I had was what? It was the switch settings that I got from mom and dad. And think about that just for a moment. In fact, my wife says, don't you ever forget to tell this part. And that is, she loves this, but it's very interesting. Think about it for a moment. So the gonads in a female, human female, are populated in the first trimester or so of pregnancy. So while our mother was in our grandmother's womb, the ovum and the cells that from which we came were produced. So it's the lifestyle of my grandmother that actually had a great deal to do with the switch settings that were in the cells that I would come from my mother's ovary. So it's quite remarkable. I don't have time to go into it, but there's been some research, interesting research done in the Scandinavian countries where they have amazing records of, and they've shown that there is a correlation between the crop production and situation during the years of puberty of the grandparents. There's a relation to how likely the grandchildren are to have chronic disease. Very interesting. So let's take a moment and look at the differences in monozygotic twins. So here, monozygotic twins are, of course, individuals that can have the exact same chromosomes. They came from a single zygote that, for whatever reason, was divided at some point in the early phase of development and became two individuals. And so I want you to look at what they did here. This was published in Proceedings in National Academy of Science in 2005. They've got, they're looking at 3-year-old twin A and B on the left, and then 50-year-old twin A and B. And what they're showing are the bands here of gene expression. And so what you notice is on the 3-year-olds, there's a little difference, but not much. But by 50 years of age, there's prominent differences in the twins that have the same genome. And what this shows is that it's the environmental effects, it's the lifestyle that changes the gene switches. The DNA in twin A and twin B is the same. But what's happening is the gene expression is dramatically changed over the lifetime. So here, a little schematic to help us understand how the switches work. So we've got there in the upper left, the chromosome, sort of this X-shaped thing. And then that's made up of chromatin. And chromatin is then a coiled DNA with also various molecules and chaperone proteins, they call it. And then if you're coming on over now to the bottom left part of this graphic, you can see there's a methyl group that's been attached to one of the base pairs. It turns out that methylation of some of the C and G base pairs is a common switch that can control, turn on or turn off gene expression. There's another switch that's illustrated here. We're only looking at two. Since this was published in 2008, we now, the number of gene switches is phenomenal. There's probably going to be more gene switches than there are genes, which is making this an extremely complex puzzle to solve. And that's why things are going slowly in some ways. But the other switch here is an epigenetic factor. This is over on the top right. The epigenetic factor is attached to a histone molecule, which is a molecule that the DNA is wrapped around. And when this tail gets this epigenetic factor, it causes these histone proteins to separate, and it allows the exposure of a segment of the DNA there. You can sort of see that in orange. Well, anyway, so this is an illustration. There's some that are much more complex than these kind of switches right here. But the bottom line is that the DNA and the chaperone proteins determine which genes are active and which ones are, okay? Which ones are quiescent or active, turned on or off. And it's interesting that as here in Nutrition 2004, Science was a publication that shows that science discovered early on that what we eat, our diet, exerts the strongest effect on our epigenetic switches. And that has still remains the case. Now, when I say the strongest, got to be clear here, especially if we have any knowledgeable critics. The fact is that when I say the strongest, I'm talking about as far as looking at the global effect on gene expression in the human body, diet seems to have more effect than anything discovered so far. There are some things that have stronger specific effects than diet does, but overall, diet is quite an amazing impact on gene expression in the body. So we've already said this, the genes are turned on and off. I already mentioned offspring. We started life with the gene switches set by the influence on our ancestors. And I explained, for example, with the development of the gonads, the ovaries in a developing fetus, how this happens. All right, I want to look at now 3 or 4 studies real quick to draw some very important points. So first I want to talk about the Agouti mouse. This is a mouse that is quite valuable, quite expensive. And there's a photo of her here on the right-hand side with the dollar signs, because what's happened is a lot of work was invested in the Agouti mouse to put genes in that would be expressed and transmitted from generation to generation that would cause these mice to have obesity, diabetes, the chronic diseases of human beings. So they've been engineered basically to be sick, so we can study these diseases and study various treatments in mice. So some researchers that were working on a little different problem, they were working on the epigenetics and about transgenerational epigenetics, were looking for a model to study to see if it was possible that diet could alter gene expression in the developing embryo, the offspring. And so they hit on using the Agouti mouse to do this research. And what they did was they fed an Agouti mouse, a pregnant Agouti mouse, a special diet that they believed would methylate the genes and turn off the genes that had been engineered into this mouse. And if so, they were expecting to see the baby looking quite different. And here is the actual photo. It's on the website for where they've done this work in North Carolina, and published in Fossab in 1998, you can see there. What we have here is these two mice. The one on the left is the adult mother, and the one on the right is the offspring, the adolescent. I often say if this was a human situation, there would definitely be a paternity suit. You know, that is not my kid. But they did the DNA testing, of course, and proved that both of these mice have the same genes for chronic disease. But the diet fed to the mother during pregnancy turned those genes off in such an effective way that the phenotype is completely missing. Even the color of the fur, which was bred in to make it easy to identify these mice, is not there. The obesity, et cetera, et cetera. And in fact, researchers found that the effect of their methylating diet were evident for 3 to 4 generations later. Of course, as a Bible student, I find that remarkable. Exodus 20 coming to mind there. But think of the implications of this. The effects of our lifestyle choices, good or bad, are passed on for generations. So I see that as a wonderful, exciting thing. And in fact, I have found this to be one of the most motivating factors for young ladies that I've ever run into in my lifestyle medicine practice, is that they can connect to this very, very well. They have a harder time connecting with the idea that when they're 50 or 60, they're going to have heart disease. All right, now the second study I want to look at here is in rats. Now these are wild rats. There's nothing special. No genes have been changed or engineered or whatever. You can see this was published in the Journal of Neuroscience in 2008. And I remember it well. It was in November 12th, because the way I learned about this paper was reading the Wall Street Journal health reporter and happened to comment on this study, because the American Heart Association was having their annual meeting, and the health reporter asked one of the acting director of the American Heart Association to comment on this study. Her comment was, it shows there's a lot more to America's obesity epidemic than we had imagined. So what is she talking about? Okay, so just quickly going through this, basically they were feeding a high-fat diet to a pregnant rat, and they were tracking what was happening in the developing embryo. And so you can see they discovered that in the developing brain of the embryo, chromosome 3, there was genes that were turned up or turned on by this diet that were causing the production of additional neurons. So just blank the screen for a moment and think what we just said. They showed that feeding the mother a high-fat diet was changing the anatomy of the baby's brain. Very interesting, but continues, it even gets more interesting. So these neurons that were being produced actually were taking up residence. They migrated to the hypothalamus. This is the type of cells that were, more of them were produced. And the result during puberty then, the phenotype, as we would call it, was that they had higher blood lipids. So they had higher triglycerides, cholesterol, et cetera. They had a higher food intake. They tended to eat more calories. They actually had a preference for fat food compared to offspring that had been born to this mother when she was fed a regular diet. So her diet actually had a positive feedback loop, making the adolescent rat want to eat fat. Had an earlier onset of puberty. These rats were able to start their families at a younger age, and they had a higher body weight. Now, any pediatrician looking at this list can tell you that that's the kind of profile we're seeing increasingly in young humans, is these very same kind of things. And so the question, of course, is how to what extent is that being caused by the high fat content of convenience food that we're more and more young mothers are eating. So now I want to look third at a study in men. This was a study published in Proceedings National Academy of Science 2008 by Ornish and colleagues. And they were looking at, it was a prospective single-arm pilot study looking at men with low-risk prostate cancer. There were 30 men in the study. These men did not have surgery or radiation treatment, which would have been more common perhaps, but they had agreed to receive a comprehensive lifestyle change treatment. Basically, they used the same program that Ornish used for heart patients. They also donated multiple or serial prostate needle biopsies. So the researchers were actually taking a needle biopsy of the prostate cancer cells at the baseline and then after the 3 months of intervention. And of course, they did not every biopsy successfully got cancer cells. Some of the biopsies were non-cancerous cell tissue, but by and large, the majority of the cases, they were able to actually get some of the tumor itself tissue. And from that, they were quantifying the messenger RNA. So they would purify the messenger RNA in very small quantities, but of course with polymerase chain reaction, they could expand that. Anyway, the point was they were able to look at what messenger RNA was present. And as you may know, messenger RNA is produced when a gene is being transcribed. The messenger RNA is transcribed and goes out into the cytoplasm where ribosome then translates it into protein. So it's a way basically to monitor what genes are active. And I won't go into it. I have a handout. You can look at these details if you're interested. It was an intensive 3-day intervention followed then by regular phone contact. They provided food so that the men could stay on the right diet. But here's the bottom line. This is what's called a heat map. And what we have here is, let me explain this so you can understand it. So on the left-hand side is pre-intervention. There's 30 columns. If you were to count them, you would find one there for each man. And on the then on the right, post-intervention, there's 30 columns, one for each man. And then on the right-hand side, there's, for each row, you might say, there's a messenger RNA that was found. And they color then at each one of those cells is light if there's very little of that messenger RNA, or very dark if there's a lot of it. So you can look at this, that's why it's called a heat map. Basically, you can look at this and see rather easily that there was a lot of change, a lot of more dark on the left and lighter on the right, but some exceptions to that. Some were light on the left and dark on the right. So when they did the count, they found that over 450 genes were turned down or off. They went from dark on the left to light on the right, but about 50 genes were turned up or on. So they went from light on the left to darker on the right. And when they looked at these genes, many of those they did not recognize, but some they did, and they recognized the genes that were known to be cancer-related, causing cancer, exacerbating cancer, spreading cancer. Those seemed to be turned down. And some of the genes that were known to be good at cancer fighting, resisting cancer, were turned up. So as Dr. Colin Campbell observed one time, wryly and quite truthfully, it seemed to me at a conference I happened to hear him, he says, isn't it serendipitous that with diet, when you make a positive change in one thing, it's positive for other things, instead of having to say, okay, I can only eat so many of these carrots to help fight my cancer before I start causing heart disease. The side effects of lifestyle, as far as we know, are positive across the whole spectrum. We call it multivalent effects. By the way, what was it these men were being treated for? They were being treated for their cancer, but look what happened. It looks like they were in a weight loss program, or no, they were in a blood pressure control program, or it almost looks like they were on a cholesterol reduction program. So this illustrates what I'm saying. They were actually having tremendous improvement in their cancer, which, by the way, was going into remission by observation and ultrasound, but it was also the genetic changes were taking place, but they had all these other positive effects as well. And here's one for me. I have to tell, my biggest challenge in life is being moderate in my work and my sleep. So this is helps me to stay motivated. They found here, this was a study you can see at the bottom done in Surrey University in the UK. 1 week of bad sleep can switch off hundreds of genes and raise the risk of a host of conditions. So they had two groups. These were 26 volunteers. These were young people, by and large, who were in college. One slept for less than 6 hours a night for an entire week. Sounds a little bit like being on call as a resident. And one that slept for 10 hours a night for that week. And then after the week of either sleep deprivation or adequate sleep, they kept these students awake for 40 hours and they took blood tests to look and see if there were effects on gene expression. And what they found was that the sleep deprivation altered the function of over 700 genes. And some of them were recognized as affecting metabolism, some inflammation, immunity, stress, et cetera. So this was done in 2013. We know now the importance of sleep and rest. Oh, here's one real quick. This was a study published in Cell Metabolism 2012. What they were asking in this study was how quickly can a exercise change gene expression in muscle tissue. And so, because some people are of the mindset that, in chronic disease, it takes years to develop, and so it must take years to reverse the gene effects. But that's not what we've discovered. In this study, they, and the bottom line is the 20 minutes. They found that within 20 minutes or less, actually they don't know how short, because that was the first measurement was at 20 minutes, but at 20 minutes, gene expression was already changing in muscle tissue as a result of exercise activity. Oops. So compare that to this little study. This was published in Circulation in 2004, and the question was asked, how do lifestyle changes compare to stenting for coronary heart disease? So they were looking at stable coronary artery disease, and it was a randomized trial, 1 year. They had exercise training in patients with stable coronary artery disease. And what was it? It was 20 minutes or more a day at 70% of symptom-free maximum heart rate. So this is very mild. They were making sure they avoided any symptoms, and a 61-hour week of aerobic group activity. Then the other 50 were assigned to get the stent angioplasty. So before I show you the results, and don't, of course, answer out loud, but think to yourself for a moment, if this was my relative, my mom, what group would I hope she got assigned to? The exercise or actually getting a stent? Well, of course, the results are pretty remarkable. Here we have what's called a survival curve, and it starts off at the baseline. All patients are event-free, but as events happen, hospitalization, angina, whatever, the curves go down, and you can see that the stent on the bottom at only 70% of those who received stents were event-free at 1 year, whereas the exercise group, 88% were event-free at 1 year. And so quite a remarkable difference. Personally, I think it's irresponsible that patients with coronary artery disease are not shown this kind of information so they can make a much more informed choice than just automatically getting a stent. So this is Dr. Feinberg, I mentioned earlier, one of the leading experts, and here's a quote from him. He says, epigenetics is at the epicenter of modern medicine. That was in his article in JAMA 2008. And lifestyle, here at the bottom, lifestyle has the greatest effect on gene expression from that publication in 2004. So put 2 and 2 together and we come up with 4. That's why lifestyle medicine is one of the fastest growing areas in medicine right now. It was mentioned earlier that I had the privilege, excuse me, of being the founding president for the American College of Lifestyle Medicine. We struggled for years to have 400 or 500 members. Now We're over 6,000 and climbing fast. It's just growing leaps and bounds. In fact, what we're just now getting into is getting the board certification in place for lifestyle medicine specialists, those physicians who practice lifestyle medicine as a specialty. It's good that we have lots of physicians that are supporting it in their primary care practice, et cetera, but we need the specialists too. So that's coming soon. All right, this is Dr. Jurtle. He's the one that did the work with the Agouti mouse. Look what I love how he says it here. He says, epigenetics is proving we have some responsibility for the integrity of our genome. Before, meaning before epigenetics, genes predetermined outcomes. Now everything we do, everything we eat or smoke can affect our gene expression and that of future generations. So epigenetics introduces the concept of free will into our idea of genetics. And I'm sure there's not a few of us who, when we were hearing all this stuff about, well, you know, you're the product of your genes, we knew this is not the true and not the full story. But until we learned more about epigenetics, we did not understand how to refute that. But now it's clear that the gene switches probably have more to do with our health or lack of it than our genes. And some people say, well, how can that be? Well, let me just tell you, the fact is that the majority of DNA changes that involve changing a base pair are deleterious. There are relatively few DNA differences that have found to be helpful. But so they're deleterious and in fact fatal in many cases. So what happens is, yes, they have a more powerful effect, but their effect is harmful enough that those people often don't survive. Of those of us who survive a normal life, the difference between our health and our disease is more explained by the gene switches than our DNA differences. So if we look at this kind of a chart, we look at health as the outcome, the phenotype that we want. What are the drivers? Well, we start at the blue there, the DNA sequence. And, you know, we have different, these numbers are not in concrete. These are expert opinion because this is questions too difficult for us to answer yet. But in general, the experts are saying somewhere in the neighborhood of 10% of the effect on whether we're healthy or not comes from the DNA sequence. And that number seems to be going down as we learn more. Medical care, whether you have insurance or coverage, whether you can see a doctor or not, you know, it's humbling to us physicians, but probably, again, only about 10% of the difference in the health status of individuals is about whether they have access to care and the environment. Here we're talking about like Chernobyl or tsunamis or whatever, that kind of thing, tornadoes may affect 10%. But the biggest chunk of it, 70 to 80%, is lifestyle, the way we live, our diet, our exercise, whether we're couch potatoes or not. And the exciting thing is we have control over that. It's not easy always to make a change, but it is changeable and it's under our control. So that's exciting. So in a quick summary, every cell in our body has a full copy of the genome. And different genes are turned on and off by environmental influences, and diet seems to be the strongest we know of so far. DNA sequence is essentially fixed, but the epigenome is stable but reversible. The epigenetic switches are covalent, so once the switch is turned on, it stays on until something turns it off. Lifestyle choices control gene expression by the epigenome. So that's why lifestyle change is an essential part of really evidence-based treatment for disease. And epigenetics is the primary science that's propelling lifestyle medicine into evidence-based medical practice. And the future is lifestyle medicine. So changing your diet and lifestyle, that can be done. And we've shown how that changes the epigenome, can even change cell type, can turn off genes in utero, et cetera. So it changes gene expression when you change the epigenome. And if you change gene expression, it changes you. So it's exciting that we have that kind of control over our bodies. So I want to thank you for the opportunity to be with you at Amen. I wish, I'm sure we all do, that we could be in person, but I'm ready to try to answer any questions I can.
John, there's a couple that were posted in the chat.
All right, let me see if I can pull up the chat.
I can just read it to you.
Okay, that would be great.
What is the difference between cell aging and body aging?
Yeah, that's a great question. So cell aging has to do with the individual cells, and body aging has to do, of course, with the entire organism. And so they're connected in the sense that we know, for example, that the cells seem to be programmed for approximately 50 cell divisions. Okay, the telomeres on the chromosomes shorten each time that there's a cell division, and that seems to be limited to about 50. So when a cell has divided 50 times approximately, that is it. It can't continue dividing. But that doesn't mean it's old. It means it just stops dividing. And by the way, one other thing I didn't mention, but when they did this GEMINAL study, Ornish did with the men with prostate cancer, they also looked at the telomerase activity, and they found, which is the enzyme that puts the telomeres back on to the chromosomes, and they found a 30-some% increase in activity of telomerase in these cells. So, so the point is, the cell aging, we have to solve that before we're going to have a significant increase in longevity of humans. But that is, and it's interesting, as a Bible student, I believe that we're not going to see 1,000-year long lifespans this side of the second coming. But I do believe that the fact we have a record in the past that human beings could live for that many years, I believe that it gives indications that there may be the possibility in our genome, in our makeup, for that kind of longevity. But anyway, so I've gone around so many places. Let me just come back to the simple answer to that question. Cell aging has to do with the cell's ability to continue functioning and dividing, etc. The aging for a human being, the organism, really is how, what shall I say, how capable, how functional we are. We know that it's not all about chronology, because there are people who are centenarians that are quite active, taking care of old people that are 30 years younger than them. And so it has a lot to do with how well we take care of the body, not just time alone. So hopefully that helps answer that question a bit.
Dr. Dant, you have a question? You want to just unmute yourself?
Yeah, I actually had a few questions. I don't know how long you're going to be here, but I'll just start with the first one. And what is a fast mimicking diet for humans? And
yes, great question, Eric. And I have that information. In fact, if you wanted to email me, oh, I'm sorry, this is an out-of-date email showing on the screen. My apologies, that's an oversight. But either Cindy or I can get you my email, and it will be on the next presentation I give later in the day. I do have the proper email there. Again, I apologize, I'm embarrassed that I've got this old email here. But I can send you that information. I can also send you the article that has it in there. But there is a fasting mimicking diet that was used with mice, and they have developed a version for humans. It's a little different. That very thought sounds good to me, and and they have used it in human beings and done some studies, published some data. So it's quite interesting, Eric, and I'd be happy to share that with you. And just for what it's worth, I have attempted it, tried it myself. Actually, I made a version of my own version because they have a proprietary product that they developed, but they do describe in the research paper basically the nutrient contents that they recommend in the caloric. What they do is in humans, it's a 5-day cycle. You have 1,300 calories the first day, and then you drop down to it's about 1,000 calories for the next 4 days. It's high in nutrients and very, low in calories is the idea. So they're trying to, with the fasting mimicking diet, they're trying to produce the benefits of caloric restriction without the negative effects of nutrient restriction. Next question.
Eric, did you have another follow-up question, or shall we go on to the next person?
I'll put mine in on the end here.
Okay. So Stuart had a question, and then there's one more in the chat after that.
Does time-limited eating, like eating within a 6-hour window as opposed to eating over a longer period of time, do the same thing as a fasting mimicking diet? Great
question, Stuart. And the fact is, so that's called TRF, time-restricted feeding. There's ETRF is early time-restricted. That's when you have breakfast and lunch and don't have supper or anything in the evening. And late time-restricted feeding has also been studied, and that's where the eating is done later in the day, in the afternoon and evening. And by the way, it's interesting for those of us who are students of Ellen White's counsels that the early time-restricted feeding has been shown to be much more beneficial to metabolism, but there are some similar effects. And now to your specific question, the thing that seems to be, because fasting mimicking diet is technically a type of time-restricted feeding, because the way they do it, they don't feed the fasting mimicking diet it's not grazing. Okay, it's specific times that you eat. So I think the fasting mimicking diet would technically be considered time-restricted feeding as well. But what I want to say is that they've done these studies in the mice, and what they showed was that there's 2 phases that are necessary for the neogenesis of the pancreatic beta cells. And that is, there has to be the fast, and then it's the refeeding is an important part of the process. It's during the refeeding that the pancreatic cells differentiate and new beta cells are produced, and during that differentiation process that happens after the fast. And so they recommend repeating 3 cycles of the fasting mimicking diet to restore, this is again in mice where they've done the most work, to restore beta cell function. So there's something more to fasting mimicking than just time-restricted feeding, but there's definitely some parallels and some interesting correlates there. Does that help answer the question?
Yes, thank you.
So the other question in the chat is, how are genetic switches related to mRNA?
Yeah, that's a good question. So messenger RNA is the mechanism. It's the way that genes which are being transcribed, which are being expressed, they if we think about our biology and what we know from our studies, we know that messenger RNA is the molecule that is created in the nucleus, and then that goes out into the cytoplasm outside the nucleus, where ribosomes, multiple ribosomes, attach to the messenger RNA and translate that into protein or whatever product it's going to be. So the messenger RNA is only produced when a gene is being transcribed. And a gene is only being transcribed or expressed when the gene switches are set for it to be turned on. If the gene switches go from turned on to turned off, then no more messenger RNA is being produced. And the messenger RNA has a half-life of hours, some minutes to hours. More in humans, more often it's a few hours. and the messenger RNA is then degraded and dissolved. So it doesn't last forever, or else the switches would be meaningless. So the messenger RNA is translated, and then it is, again, disappears. And so gene expression has to then be turned back on at some point for that same messenger RNA to be produced again. Does that help explain the relationship?
Yes, thank you.
Good.
Eric, did you want to go ahead and ask your other question?
Yeah, and maybe just a follow-up to the messenger RNA, and maybe this was part of the question originally, but do you have anything to speak to in regards to the experimental vaccines and the messenger RNA in this context?
Well, I will say that, I mean, since this can be a controversial subject, but I believe that we can talk sensibly and ignore, hopefully we can ignore all the political baloney and talk strictly about science here. So yes, there's some interesting parallels. So when messenger RNA is produced inside the cell, as we've been talking, that's when a gene is turned on and messenger RNA is produced and then goes into the cytoplasm and so forth. Well, with the messenger RNA that's being used as a vaccine, it's very interesting. It's in some ways, it's almost like it's not a vaccine, and it's certainly not in the normal sense of a vaccine, because we typically think of a vaccine more like the Johnson & Johnson or the smallpox or polio vaccine, where we have taken the virus and we have weakened it, or taken a piece of it and inject that into the human being so that our immune system then recognizes this foreign material and builds up an immunity. With the messenger RNA vaccine approach, what we're doing is we've developed a way to make a messenger RNA molecule, put it in a capsule that will can be injected into the body and be taken up by cells. And when this messenger RNA then is released into the cytoplasm, the ribosomes treat it like any other messenger RNA. They start producing the proteins that it codes for. And the proteins that it codes for have been created in the laboratory. They didn't come from the virus. I mean, you know, the sequence was developed based on knowledge about the proteins in the virus. But the point is, none of that material actually came from a virus. It was created as a copy. And I won't go into great detail, but there was a challenge for many years. This was unsuccessful because it took them a while, these amazing scientists, a while to find the right key to changing the messenger RNA to make it so that when it was taken up by the cells, it would function in a normal, non-harmful way. Having said all that, people ask me sometimes, I'm sure they ask you too, if are you going to take the vaccine, and which one are you going to take? And it's just my own, you know, I'm not going to tell anybody what to do. But for me, I'll take the old style design. I'm more comfortable with the old design until we have a few more years of experience with this new approach. I think it may be wonderful, and maybe part of the reason for my caution is because I'm old. Some of my young friends tell me, well, you're just too old, try new stuff. But I am cautious, and I think that I would be a lot more comfortable if we had a few more years of experience. But at any rate, I think it's an exciting development that we've got, and I hope that it all goes very well.
So a couple of other things that have been put in the chat is regarding handouts. And also if you could send the fasting mimicking diet to more than just Dr. Dant.
Sure, why don't I just do this? Why don't I send it to you, Cindy? Can I do that? Do you have everyone's? And you can just send it, yeah. And anybody who didn't want it can just delete it.
That would be fine. I can send it out to everyone who has registered for the conference once it's over.
Sounds good.
Are there any other questions?
How much time do we have left?
Well, it depends on whether you want to break or not.
Yeah, I'm happy to answer questions, but I think we're getting into the break time. But whatever you do, I love this stuff.
Perhaps I can email you, but just right offhand, you mentioned the female component, genetic component, being critical, but did not speak to the male component. So I'm just wondering if there's any genetic consequences there as far as epigenetics and offspring and grandkids. The other interesting thing is, as you know, that meat was a very important part of the Jewish economy and is almost compulsory to eat the sacrifices and eat meat back then. I understand that there were some significant differences in the meat itself and in human beings, and that it wasn't a constant diet, but I'd be interested to know your comments on that as well. But again, yeah. I can do this through email as well.
Sure. Well, let me just speak. I think the one about the Jews and the meat and the differences, that's a long, I would be happy to have that conversation by email. But the one that you asked, I think it's very relevant. I should have spoken to about the male effect on the epigenome. So yes, there are indeed, of course, let's go back to a little science first. So in the case of the male gametes, these are produced in the testes on an ongoing basis, as we all know, that unlike the female ovaries, which are populated and then that's it, they begin life with a finite number of ova. In males, we produce sperm, as far as we know, for an entire lifetime. And so that means there's a number of factors that play into that. What that means is that theoretically, the effects of my lifestyle on the gene switches of my gametes, of course, would be only on the last, month or so, maybe only a couple weeks, because the gametes are being produced, right, and recycled. So when I have a couple of children, so when my children were fertilized by my sperm and their mother's ova, that was sperm that probably wasn't more than a month old, right? So that would mean that theoretically my lifestyle effects bequeathed to my children were based on the more recent past than with their mother. The mother, the effects were from as far back as in the womb. Okay, so that's one thing I want to explain. There is a difference there in the time frame, but one of the ones that have been shown to be very significant, believe it or not, is smoking. There's published papers showing that the gene switches on the bequeathed to the developing embryo by the father, biological father, are affected by smoking and measurable. And they're not beneficial, by the way. And so it's a lot more work to be done if we want to really find out all the effects. But there are effects been documented from the father, from the male, transmitted through the sperm, but those changes are not, like I say, don't go back as far. Those effects seem to be more recent. As far as the question about eating meat and so forth, I would say I don't think we have any good way to know what the meat was like or food was like that many years ago, even back to the Jews in Christ's time. I'm aware and fully accept the fact that Jesus ate fish, and I don't know that he didn't eat I assume he ate other things. I presume he ate lamb and whatever else that Jews ate. But that's not the issue as far sticking with just the science. Right now, we have some very, very solid science that tells us a whole food plant diet it seems to be the best dietary plan for longevity and healthy function in the majority of people. Now, there's a number of qualifiers there. I am well aware, as anybody who does a lot of lifestyle medicine treatment, that all people don't have the exact same response to dietary or other lifestyle things. And it appears that I think that's to be expected, frankly. But anyway, it appears that there are some people who do better on a more Mediterranean-like diet than on a strict low-fat plant diet. I'm not sure I have seen any indication that there are people who do better on a high fat, high animal food diet. I know there are advocates of it, but my own take, for example, let's take the so-called ketosis or ketotic type diets that sort of come into popularity and then wane and then come back. And I think we're on a cycle right now where they're sort of in the middle. But anyway, that research showing, for example, improvement in weight control or glucose is not, the published studies do not show long-term results yet. They've not done the work that we need to look at, and there's every reason to believe that the higher cholesterol and higher saturated fat levels are going to exert their normal proven effect on blood vessels, atherosclerosis, etc. So until we can see some results that somehow you can avoid those documented proven ill effects of high animal food, high saturated fat diet using a ketosis diet, I'm not ready to buy into it. However, if I have patients or friends that are on it, and I say, "How's it going?" They say, "Well, I'm helping. My weight's down and my blood sugar is better." I say, "Well, let's hope it works." My biggest concern as a scientist is that there may be ill effects that you'll regret later. But anyway, most of these people become very attached to it, and it's a little bit like the Bible says, let them grow together and we'll separate them at the harvest. Sometimes people get so excited about a diet that it's not worth trying to take it away from them.